Thank you for the introduction. Good morning, everyone. My name is Jordan Allen. I'm a senior here at North Central College, graduating in the next few weeks. That is, if I can make it that long. And I will be presenting on my thesis on quantifying reactive astrogliosis during prolonged forced abstinence from oxycodone self-administration. I know that's a mouthful and it sounds like an awful title, but hopefully by the end of this presentation, you will understand that it is the most comprehensive summary of this research that I could put into a title. Let's get started. So before we jump into the specific research, I want to provide a broad sense of why we conducted this research in the first place. Now, most of you know by now that there's an opioid epidemic taking place, and this is largely due to the overprescription of opioids in recent years. Oxycodone is a semi-synthetic opioid, and opioids in general are just a class of pain-relieving drugs. And opioids are a huge problem right now because they're causing millions of individuals to lose their lives to an addiction. Some frightening statistics here: approximately 27 million people worldwide are currently suffering from an opioid use disorder, and roughly 100,000 opioid overdoses occur each year. I also want to make sure, before we get started, that everyone understands what drug addiction is. It's defined as the brain disease characterized by compulsive drug seeking despite negative consequences. And what's important to realize here is that drug addiction is a brain disease. It may start off as a choice to take drugs, but once an addiction develops, it becomes a necessity. Having spoken to hundreds of injection drug users myself, most of them crave a life without drugs, but just cannot seem to abstain from them no matter what they do. So among the primary drivers of this opioid epidemic is a lasting vulnerability to relapse, which is the largest issue with treating drug addiction. And part of this lies in the fact that there are some pretty nasty withdrawal symptoms from opioids, such as insomnia, anxiety, cold sweats, nausea, much more. In the top right, we can see the timeline of these symptoms, which peak after 72 hours and subside after about a month. Interestingly, though, people are vulnerable to relapse even after their withdrawal symptoms disappear. And this may be due to a phenomenon known as the incubation of drug craving, which is defined as the time-dependent intensification of drug craving during withdrawal. And the incubation of drug craving has been seen in humans with nicotine addictions and in rats with a variety of drugs. And we can see this demonstrated by the figure in the bottom right, where we can see that the rats pressed a lever that gave them infusions of cocaine more and more the longer they were in withdrawal. So most research on drug addiction up to this point has focused on the neural adaptations, or the structural and chemical changes that take place in neurons. However, researchers are beginning to understand the critical role of glial cells, which are the support cells for neurons. Astrocytes, shown here, are the star-shaped glial cell, and they have projections to neurons and blood vessels. They're the most abundant type of glial cell. In fact, they outnumber neurons, and they seem to be critically involved in maintaining substance use disorders. They mainly function to modulate neurotransmission, maintain glutamate homeostasis, and provide neurons with nutrients. After an injury or some other traumatic insult of the brain, astrocytes can undergo a process called reactive astrogliosis, which is a description of active astrocytes that is characterized by astrocyte proliferation and hypertrophy, which is the enlargement of astrocytes. And they are also characterized by the secretion of cytokines, which are inflammatory substances that are involved in the immune system, and they are pro-inflammatory, meaning that they trigger neuroinflammation. So there are two main types of reactive astrocytes: the A1 astrocytes, which are inflammatory and neurotoxic, and the A2 astrocytes, which are beneficial. The A1 astrocytes lose almost all of the normal astrocyte functions, and such as their ability to promote neuronal survival, outgrowth, and synaptogenesis, the creation of synapses. And importantly, they induce cell death, which explains why they're implicated in neurodegenerative diseases such as Alzheimer's. For this presentation, what you should remember is that A1 astrocytes are marked with C3, which is a complement cascade protein involved in immune responses. A2 astrocytes, on the other hand, upregulate many neurotrophic factors that promote neuronal survival and serve to protect neurons. GFAP, or glial fibrillary acidic protein, is an intermediate filament protein and it's primarily expressed in astrocytes, and it's responsible for providing the structural support to astrocytes and assisting with shape changes and movements of the astrocytes. And C3, again, is our marker of A1 astrocytes. Previous research has shown increases in GFAP after exposure to opioids. And what I want you to remember is that an increase in GFAP is indicative of astrogliosis. And so using this information from previous research, we hypothesized that as a compensatory response to no longer having oxycodone, we would actually see decreased GFAP and C3 levels on withdrawal day 1, and an even larger decrease in GFAP and C3 on withdrawal day 15 as the traumatic insult of the exposure of oxycodone is further removed. So in order to assess the craving of oxycodone, we first had to train the rats to self-administer oxycodone. So to do this, we put the rats in operant chambers for 6 hours a day for 10 days. And while they're in this chamber, the rats could either press the active lever, which would result in an infusion of oxycodone paired with a light, or they could press the inactive lever, which did nothing. And then following this self-administration training, the rats remained in their cages for 1 or 15 days while they underwent withdrawal, otherwise known as forced abstinence, because the rats are being forced to abstain from the drug. Okay, so what do we find? So looking at this figure on the top left, we can see on the x-axis that we have the day of self-administration training, and on the y-axis, we have the number of lever presses, either active or inactive. So in the oxycodone rats, the top left figure, as the self-administration training progressed, the number of active lever presses increased, which is shown in red, while the number of inactive lever presses, the lever that did nothing, remained the same, and this is shown in the white bar. So the rats in the oxycodone condition learned that they would receive an infusion of oxycodone with an active lever press, so they significantly preferred that lever over the inactive lever as training progressed. Rats in the saline condition, which just received a saline infusion, which is not rewarding, did not learn to distinguish between the levers and did not develop a preference towards either lever. Then we wanted to assess the incubation of drug craving, or the intensification of drug seeking over withdrawal. And so to do this, we recorded the number of active lever presses in a 30-minute drug seeking test. And rats that preferred the active lever or press the active lever more were said to have an intensified drug craving for oxycodone. And so going through the various elements of this graph, we have the various withdrawal days in the saline and oxycodone conditions on the x-axis, and on the y-axis, we have active lever presses. So between the oxycodone and saline animals, we found that at both time points, oxycodone animals press the active lever significantly more than the saline animals. Now, just looking at the oxycodone rats, we can see that the active lever presses, shown in red, were significantly higher on withdrawal day 15 than they were on withdrawal day 1, which is the gray bar. And so this indicates that the craving for oxycodone intensified or incubated over the 15 days of forced abstinence. And since we love neuroscience, we had to look at their brains during this incubation of drug craving. And so on the left, we can see immunofluorescence images of the medial prefrontal cortex and the nucleus accumbens on withdrawal day 1 and withdrawal day 15 in the oxycodone rats. And so the red markings that we see show us GFAP, which, as I mentioned earlier, indicate astrocytes generally. So the red markings show us astrocytes. And from these images, it looks like astrocytes are more abundant in both brain regions on withdrawal day 15 compared to withdrawal day 1 of the oxycodone rats. But when we quantified these GFAP markings based on their overall area, which we can see in the figures on the right, we found that this pattern of slightly increased GFAP levels on withdrawal day 15 compared to withdrawal day 1 was not significant. Okay, so what about A1 astrocytes, the cool inflammatory A1 astrocytes? So the top row here is showing one spot in the medial prefrontal cortex, and the bottom row is showing one spot in the nucleus accumbens across that row. And looking from left to right in each row, we have the first one being GFAP, which is, again, to measure astrocytes generally. The second one, the middle bar, shows the C3, which was to measure A1 astrocytes. So everything you see here is potentially an A1 astrocyte, and a merged image all the way on the right to show where GFAP and C3 overlap or co-localize. So the important finding here is that there were markings when we stained for C3, which we can see in the middle and the white dots. And there is some degree of co-localization in both the MPFC and the nucleus accumbens, which was expected since A1 astrocytes are transformed astrocytes. And so this increase in C3 tells us that A1 astrocytes, the inflammatory astrocytes that cause cell death, are present in both the medial prefrontal cortex and the nucleus accumbens during withdrawal or during the incubation of oxycodone craving. So again, the major findings: one, we were able to successfully induce the incubation of oxycodone craving, meaning that the craving for oxycodone did intensify during withdrawal. And there was a consistent but non-significant pattern of increased activity or increased astrocyte levels on withdrawal day 1 compared to withdrawal day 15, compared to withdrawal day 1 in the oxycodone animals, which, as I mentioned earlier, is indicative of astrogliosis. So if these trends were significant, they would tell us that there are more astrocytes in both the medial prefrontal cortex and the nucleus accumbens the longer the rats were in withdrawal. And so the last important finding is that A1 astrocytes are present in both the medial prefrontal cortex and the nucleus accumbens of oxycodone rats on withdrawal day 15. And the reason this is such an important finding is that it indicates that there is cell death occurring during the incubation of oxycodone craving or the withdrawal of oxycodone. How exactly A1 astrocytes contribute to the incubation of drug craving? We still don't really know, since we only were able to examine the brains of rats on withdrawal day 15. And for this reason, we didn't have a withdrawal day 1 time point to compare withdrawal day 15 and withdrawal day 1. So we can't see how the levels of A1 astrocytes are changing over time. So we can't make any conclusions based on like what they are exactly doing. But this research just shows that they are present. So future research should focus on this more. And so better understanding A1 astrocytes can have profound impacts on the field of neuroscience. A1 astrocytes seem to be involved in neurodegenerative diseases such as Alzheimer's and traumatic brain injury. So understanding their function in response to drug exposure will help us understand their roles in many other conditions. And hopefully, with an improved understanding of the neurobiological mechanisms of drug addiction, we can help to prevent the loss of lives due to drug addictions.
Thank you, Jordan. I presume you can hear the uproarious applause